Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine your brain, or maybe even a tiny fruit fly's brain, pulling off these incredibly precise, super fast actions, like, dodging something that suddenly pops up.
Right, lightning fast.
Yeah.
Right.
And we often think of brain wiring as, well, fixed, don't we? Like a rigid network, like electrical circuits.
That's the classic view.
But what if the brain actually uses something much more fluid, maybe something like a dimmer switch to tune those connections, to make sure every action is just perfectly calibrated?
A dimmer switch, I like that.
Well, this deep dive explores this really groundbreaking idea, how your brain might literally paint its connections using these subtle molecular gradients, painting them to translate what you see into exactly the right action every single time.
That's a great way to put it.
Yeah.
And yeah, today our deep dive is unpacking a truly significant study. It was published in Nature just this year, 2025. The title is Molecular Gradients Shape Synaptic Specificity of a Visuomotor Transformation. So our mission really is to navigate through this pretty dense scientific paper.
Yeah.
We want to pull out the most important insights so you can really grasp how even these incredibly subtle molecular differences can profoundly impact how brains get built and crucially how they function.
Right, get those aha moments.
Exactly. The goal is for you to walk away feeling genuinely well-informed about this really cutting-edge research.
Okay, let's dive in then. So our brains, they're constantly turning what we sense into what we do.
Constantly.
Think about catching a ball, or like we said, for the fruit fly, suddenly dodging a shadow, a predator maybe.
Yeah, that looming threat.
And this requires just almost unbelievable precision in the neural wiring. You need the right cells connecting in exactly the right places and with exactly the right strength. It's mind-boggling.
It really is. And for a long time, we've known about these things called guidance molecules. Think of them like signposts. They steer the nerve cell extensions, the axons, to the generally correct regions in the developing brain.
Toward the right neighborhood.
Yeah, exactly. The right neighborhood. But the really deep question, the kind of murky part of the map has always been, how do these subtle molecular cues establish synapse-level specificity?
Okay, so not just the neighborhood, but the specific address.
Precisely. How do they control the exact number and the strength of connections within those broad regions, especially in these really critical circuits that transform vision into movement? And this paper, well, provides a really stunning and elegant answer.
So where did they start? What system did they look at?
They started by focusing on this perfect little natural system in the fruit fly, its escape circuit.
The dodging response.
Exactly. It's triggered by what scientists call looming motion, basically, a dark object rapidly getting bigger in the fly's view, like a predator swooping in.
Okay, makes sense.
And this immediately triggers an escape, a really fast takeoff.
So which specific neurons are involved?
They honed in on a particular type of visual neuron called LPLC2. Its job is detecting this looming motion. Then they looked at how LPLC2 connects to another crucial neuron, the giant fiber, or GF neuron.
The GF, that sounds important.
Oh, it is. The GF is like the fly's central command for triggering that super fast escape takeoff. It's the direct link from seeing the threat to taking action.
Okay, so how do these LPLC2 detector neurons talk to the GF action neuron? Is it just a simple connection?
Well, this is where it gets really interesting. They use these incredibly detailed 3D reconstructions of the brain's wiring.
Like a circuit diagram?
Kind of, but way more complex. Imagine mapping every single connection. And doing this, they stumbled upon something completely unexpected. These LTLC2 neurons form what they described as a dorsoventral synaptic gradient with a GF.
Dorsoventral. Synaptic gradient. Okay, break that down.
Right. So in plain English, it means the LPLC2 neurons that detect looming motion coming from the fly's dorsal visual field. That's the upper part.
Okay, looking up.
They form significantly more synapses, more connections with the GF neuron than the LPLC2 neurons detecting motion from the lower visual field.
Wow, so it's not the same connection everywhere.
Not at all. It's beautifully graded, like a brushstroke that's heavier at the top and fades towards the bottom. This synaptic gradient just refers to how the number of connections isn't random. It varies precisely along this top-to-bottom axis.
So it really is like that dimmer switch analogy, more juice from the top.
Exactly like a dimmer switch. It's a connection dimmer switch.
That is a profound discovery just on its own, but what does it actually do for the fly? I mean, if the upper neurons connect more strongly, does that mean the fly reacts differently if the threat comes from above versus below?
Precisely the question they asked.
Yeah.
And they tested it directly. They showed flies these looming stimuli, but at different elevations, and the results were crystal clear. A strong behavioral correlation. Flies responded much more strongly. They used more of those GF-driven short mode takeoffs, which is their absolute fastest escape mode.
The panic button.
Pretty much. They hit the panic button more often when the stimulus came from the dorsal, the upper visual field.
Okay, that's direct proof, though.
Absolutely. It's direct, compelling evidence that this LPLC2 GF synaptic gradient, this difference in connection numbers, generates a corresponding gradient of GF activation. It literally shapes the fly's escape behavior based on where the threat is coming from.
Wow.
It's a beautiful direct line connecting the subtle details of synapses to a very precise, observable action.
Okay, so we have this elegant synaptic gradient, this dimmer switch shaping behavior, but the deep mystery is still there. What's the molecular blueprint? What's the instruction manual for building such a precise graded connection in the first place?
Right, and this is where their truly innovative approach shines. They use a technique called single-cell RNA sequencing, or cRNA-seq.
Okay, I've heard of that, looking at genes in single cells.
Exactly. It lets you look at the gene expression, which genes are turned on, and how strongly in individual LPLC2 neurons. And they did this across different developmental stages.
So like getting a unique recipe card from each cell.
That's a perfect analogy, peeking at each cell's internal recipe card to see what ingredients it's using.
And what did those recipe cards tell them? Did they find like two distinct types of LPLC2 neurons, a dorsal type and a ventral type?
That's what you might expect, right?
Yeah.
But what's truly fascinating here is that it wasn't discrete categories at all. No. Instead, they discovered a continuous gradient of gene expression across the entire LPLC2 population.
A smooth change.
A smooth, continuous change. They identified key players, these cell recognition molecules. Think of them as molecular handshake proteins like Dpr13 and DIP-6.
Okay.
And here's where the dimmer switch analogy really hits home.
Okay.
There was no clear on-off boundary. Just this smooth, continuous change in the expression levels of these molecules, shifting incrementally from one end of the LPLC2 population, the dorsal end, to the other, the ventral end.
Wow.
This underlying molecular tapestry, this difference in gene expression levels or transcriptomic heterogeneity, forms a genuine, continuous molecular gradient.
So they saw the instructions, the RNA levels, formed a gradient. But could they actually see the molecules themselves laid out like that in the neurons, following the gradient physically?
Yes, they could. And this is where the visual proof comes in, making the picture really complete. They use these advanced imaging techniques. One is called HCR-SI-RIH,
Okay.
which literally lets them visualize and count individual mRNA molecules right inside the neurons. And using this, they confirm these gradients visually.
They could actually see more Dpr13 in the dorsal ones.
Exactly. They could see that LPLC2 neurons, whose dendrites the input branches were in the dorsal visual field, had higher levels of molecules like Dpr13 and beat variety mRNA. And conversely, the ventral neurons had higher levels of other molecules like CAT and Dpr17.
So the molecular blueprint perfectly matched the anatomical map.
Perfectly. It provides powerful validation. It's like checking the painter's palette and seeing the colors really do match what's on the canvas.
Okay, now we're getting somewhere deep. They've found these molecular gradients. They've seen them expressed in the right places. The million-dollar question now is, do these molecules actually cause the synaptic gradients? How do these tiny molecular dimmer switches work to build those specific connections?
Right, causality. That's the key. And it raises an important point. If the LPLC2 neurons are expressing these graded recognition molecules,
The handshake molecule.
yes, then their partners on the other side of the synapse, like the GF neuron, must express complementary binding partners, something for them to shake hands with.
Makes sense. Did they find one?
They did. They identified a molecule called DIP as the key binding partner for Dpr13, located on the GF neuron.
DIP-epsilon. Okay.
So think of Dpr13 on the LPLC2 neuron as the maybe synapse builder, and DIP on the GF as its perfect receptor lock. They need to find each other and click together to make a strong connection.
The molecular handshake. So if that handshake is critical, what happens if you mess with it? If you break that connection between the builder and the lock?
Excellent question. They did exactly that. They engineered flies where DIP was either removed entirely or significantly reduced, specifically in the GF neuron.
And the result?
The results were really striking. They saw a dramatic reduction in the physical overlap between the LPLC2 axons, the output parts, and the GF dendrites, the input parts. And crucially, fewer synapses actually formed between them.
Fewer connections?
Way fewer. This directly showed that DIP is absolutely essential for establishing and maintaining that precise LPLC2-GF synaptic connectivity. It's a clear causal link. Without that molecular handshake, the connection just doesn't form correctly, or at least not as strongly.
Okay, that's the physical connection. But did this disruption translate to changes in the fly's actual escape behavior? Did breaking that molecular handshake mess up how the fly dodged threats?
Absolutely it did. The loss of DIP in the GF neuron led to significantly weaker responses in the GF itself.
Less activation.
Less activation, and consequently a dramatic reduction in those super-fast short-mode takeoffs. And this is true regardless of where the looming stimulus appeared, top or bottom.
So it weakened the escape response overall.
It did. But here's the really powerful part, the clincher experiment, in my view. They did the opposite, too. They overexpressed Dpr13, the synapse builder, uniformly across all the LPLC2 neurons.
So instead of a gradient, they just turned all the dimmer switches to full blast.
Exactly. They completely flattened the natural Dpr13 gradient. And what happened to the behavior?
What?
The flies responded uniformly to both dorsal and ventral looming stimuli. The naturally graded escape response, the difference between reacting to threats from above versus below, was completely eliminated.
Wow. That is definitive.
Isn't it? It's a stunning demonstration. Manipulating that single molecular gradient causally altered both the number of synapses and the precise context-dependent behavior. It's direct proof that the gradient itself is the key functional element.
Okay, so Dpr13 and his partner DIP-ε, they seem to be the main conductors for the output side of LPLC2, controlling how strongly it signals the escape command. But you mentioned another molecular gradient earlier, beat6.
The memory, yes, beat6.
Did it play a similar role or something different?
Well, beat6 also showed that distinct dorsoventral gradient in LPLC2 higher expression up top, just like Dpr13.
Okay.
But they hypothesized this molecule might be doing something different, maybe controlling the input side of the LPLC2 neurons.
How they receive information.
Exactly. Specifically, how they receive information from other motion-detected neurons called T4-T5 neurons.
T4-T5, what do they do?
They're specialized. For example, some subtypes like T4D and T5B are specifically tuned to detect downwards motion.
Ah, okay. So how did this beat6 gradient affect how LPLC2 gets information about motion, maybe specifically about downwards motion?
Well, they found that beat6 interacting with its partner molecule called SIDE2, which is found on those T4D, T5D neurons.
Another handshake.
Another specific handshake. This beat6 SIDE2 interaction directly sculpts the dendritic branches of the LPLC2 neurons. Remember, dendrites are where neurons receive their inputs, like antennas.
Right, so beat6 shapes the antenna.
Precisely. When they knocked down beat6 in LPLC2, they saw a graded loss of these critical dendritic branches, especially in the dorsal LPLC2 neurons, the ones that normally have high beat6.
So the antenna got smaller, especially up top.
You got it. And conversely, overexpressing beat6 had the opposite effect. It increased dendritic branching, particularly in the ventral neurons that normally have less. So beat6 isn't just a passive signal, it's physically shaping the neuron's structure to receive specific kinds of information.
Okay, structure changes.
Yeah.
But what did this mean for how the LPLC2 neuron itself actually sees or perceives motion? How did reshaping the antenna change its function?
This is where the functional impact of this second gradient becomes really clear. In a normal wild-type fly, the dorsal LPLC2 neurons are sensitive to both upwards and downwards motion. They get input about both.
Okay.
But the ventral LPLC2 neurons are predominantly sensitive only to upwards motion. Why? Because they naturally receive less input from those downwards detecting T4D, T5D neurons, partly because they have less beat6.
Right, weaker handshake for downward signals.
Yeah, exactly. So when they knock down beat6 in the dorsal LPLC2 neurons,
The ones that normally see both up and down motion.
yes, those dorsal neurons suddenly became biased towards upwards motion only. They started behaving just like normal ventral neurons.
Whoa!
Yeah, it shows that this beat via SIDE2 molecular gradient precisely tunes the directional sensitivity of each LPLC2 neuron based on its position in the visual field. It fundamentally influences how they integrate different motion cues before deciding whether to signal danger.
So it's this beautifully layered system. One gradient, Dpr13, controls the output strength, and another gradient, beat6, fine-tunes the input sensitivity.
You've got it. Two different molecular dimmers controlling different aspects of the same circuit's function.
Okay, this is genuinely a monumental leap in understanding. We've gone from tiny, subtle molecular differences all the way to precisely shaped escape behaviors. What's the absolute biggest takeaway here? The core innovation that this study really brings to the table for how we think about brain wiring.
I think the conceptual advance is just huge. It moves us so far beyond simply mapping broad regions of the brain, blobs on a scan. This study reveals synapse-level specificity, that incredible precision right down to individual connections.
Its specific address.
The specific address. And it shows this specificity is directly encoded by these graded molecular signatures. It offers this quantitative, really elegant model. You have a continuous gradient of gene expression that leads directly to a corresponding gradient in how likely synapses are to form and how strong they are.
Okay.
Which then scales up to produce a graded sensorimotor gain, a graded behavioral response. It essentially tells us how the brain constructs these incredibly precise functional maps.
So yeah, it's not just getting neurons into the right zip code anymore. It's about giving them that exact street address, maybe even the apartment number, telling them who to connect with and exactly how strongly, all to achieve a specific function.
That's a perfect way to put it. And this research, it just elegantly closes this long-standing gap we've had in neuroscience. It directly links those developmental guidance cues, these molecular gradients that are laid down as the brain is built, to how adult circuits actually function and produce complex, nuanced behaviors. It's a truly comprehensive sort of end-to-end explanation.
And you mentioned the methods. It sounds like pulling this off was a technical feat in itself.
Oh, absolutely.
Yeah.
This study is also a methodological triumph. It beautifully integrates spatial transcriptomics, seeing where the genes are active, with targeted genetic perturbations messing with specific molecules.
Right, the knockdowns and overexpression.
Exactly. And functional connectomics, mapping the actual connections and seeing how they work. That's an end-to-end evidence chain, going from the gene expression patterns all the way to the observable behavior. That's incredibly rare and really difficult to achieve convincingly in a single study.
So connecting this to the bigger picture then, does this principle, this molecular gradient idea, likely apply elsewhere?
That's the exciting part. This framework seems remarkably scalable. It strongly suggests that similar graded molecular codes could be operating in many, many other topographic systems throughout the brain.
Like in humans?
Very possibly. Think about how our own visual cortex processes information from different parts of our visual field, mapping it precisely. Or how our motor cortex controls different muscles with fine-tuned strength. It's highly probable that these kinds of precise, dimmer switch-like molecular gradients are at play there too, even in much more complex mammalian visuomotor loops, including our own.
This is truly mind-bending stuff. It just makes you wonder what else is being controlled by these subtle molecular variations under the surface. So what are the big open questions now? Where does this research lead? And maybe down the line, what could it mean for us, perhaps in areas like medicine?
Yeah, great questions. Open challenges certainly include understanding how these genetically programmed molecular gradients might interact with experience-dependent plasticity. How do learning and memory fine-tune or maybe even resculpt these pre-wired gradients over time?
Nature versus nurture meeting at the molecular level.
Kind of, yeah.
Good going.
But I think the real potential game changer lies in the application possibilities. This framework suggests really actionable strategies down the road. Yeah. Imagine, perhaps, the possibility of reimposing or mimicking these specific molecular gradients therapeutically.
How so?
Well, maybe to restore precise connectivity after a brain injury, like a stroke. Or perhaps to rebalance pathological sensorimotor gains in neurological diseases where circuits are maybe over or underactive.
Like Parkinson's or something?
Potentially, or conditions involving sensory processing. It offers a completely new way to think about repairing brain circuits, not just trying to stimulate broadly, but intervening with astonishing, almost surgical molecular precision.
Wow. Okay, so wrapping this up, what does this all mean for you, the listener, thinking about the incredible biological machine inside your own head? We've seen how this elegant, continuous variation in just a few specific molecules, expressed not just as on-off, but as a true gradient, a dimmer switch can precisely wire up neurons, shape exactly how strong their connections are, and fine-tune complex behaviors like a fly's lightning-fast escape.
Mhm.
It's just a powerful reminder, isn't it, that sometimes the most subtle, continuous differences at the molecular level can have the most profound and incredibly precise effects on how we perceive and interact with the world around us.
It really is, and it definitely raises an important follow-up question. If our brains and other animal brains build such precise functional maps using these graded molecular addresses or dimmer switches, what other subtle, continuous molecular variations might be at play that we haven't even discovered yet?
Mm.
Could they be shaping everything from our individual perception of reality, maybe why colors look slightly different to you than to me, to our unique cognitive abilities, or even influencing conditions we're still trying to understand, like autism or ADHD? It just prompts us to consider this vast, hidden molecular orchestra that might be conducting our neural symphony, constantly fine-tuning our experience in ways we're only just beginning to appreciate.
So next time you effortlessly catch something you dropped, or maybe you react incredibly quickly to something you glimpse in your peripheral vision, just pause for a moment. Could it be that your brain isn't just relying on rigid, fixed wiring like we used to think, but maybe on a beautifully orchestrated molecular gradient, a finely tuned dimmer switch that allowed for that lightning-fast, perfectly calibrated response? Something to think about. Keep those wheels turning, and we'll be back with another deep dive soon.